Hello and welcome to the Cancer Drug Development Forum webinar on Bayesian approaches in drug development. In a short moment, we will hear Rob Hemmings speak about this topic. And very welcome. We have a very high attendance today on this summer day in July, and we are very happy to welcome all of you to the CDDF. Please, the next slide. The Cancer Drug Development Forum is a non-profit organization registered in Belgium that provides a neutral platform to stimulate interaction between stakeholders involved in cancer drug development. We bring together leading voices from academia, pharmaceutical industry, regulatory authorities, policymakers, and patient advocacy groups to improve cancer treatment. Please check our website, cddf.org, for our programs, which are open to all interested people. Please, next slide. We have a rule that is mainly related to the discussion, as the webinar itself will be published. The Chatham House rule, so you're free to use information received, but not so much to share the identity or affiliation of the speaker. And that is also true for social media. So if you want to quote someone, you have to get the permission of this person. Some technical aspects in the next slide. So if you have any technical problems or something like that, please use the chat function. You see an icon on your Zoom bar on the lower part of your window. And that is the way to tell us that you have some problems with sound or video. The Q&A function is for the questions, and you can put in questions at any time. We are very happy to do that. And we will, in the discussion part, work through these questions. You also have the opportunity to vote these questions up if some questions are more interesting to you than others. So these questions will be given to Rob during the Q&A session. And you can double-click on the Zoom screen to enter full or exit full screen mode at your convenience. Today's program is on the next slide. My co-chair, Professor Eva Skovlund, who is a proper statistician, and I will moderate the discussion, and our speaker will be Rob Hemmings. And the topic are the Bayesian approaches in drug development. So what is that about? Let's look at the next slide. It is about the probabilistic thinking in statistics, I would say. So Thomas Bayes, a referent in England, and this is the only probable picture of him, is seen as the founder, but there was also a Frenchman, Pierre-Simon Laplace, who has published about it. So these two people have introduced a different way of thinking in statistics than the usually frequentist statistics, which we use with p-values, confidence intervals, and zero hypothesis thinking. So I was not trained in this in my medical studies, so I looked to the literature available to my level of medical understanding. Please look at the next slide. And I consulted this instructive book, and it helped me a lot, actually. Next slide. Because it asks the question, there are cookies with and without pieces of candy. So does this sample that we see here come from one with candy? How do I answer this question? And please go forward. Here, very simply, you look at the prior distribution of cookies, and then you can decide how in the posterior distribution of cookies, how likely it is. And there are many important applications to this, but we will hear much more on a much higher scientific level from Rob, who is, please, the next slide, who is a very experienced statistician. He has worked at the CHMP, and he has chaired the Scientific Advice Working Party for 8 years. He chaired expert groups on biostatistics, modeling, simulation, extrapolation. He has authored many regulatory guidance documents, and now he is working at a consultancy together with his colleague Thomas Samuelsson. He has a particular interest in when and how to use data in clinical practice to support drug development, and he is co-author of a very interesting article to which he will refer in his talk, which gave us the idea to convene this webinar. So enough said for introduction. I'm very happy to see that we have now 136 attendees, and Rob, I turn it over to you for your presentation.
Hello, everybody. My name's Rob Hemmings, and it's a pleasure to be here today to give an introduction to Bayesian approaches in drug development. And thank you to Axel, Eva, and the CDDF group for the opportunity to be able to present. Here's a short disclaimer. I currently work as a consultant to pharmaceutical industry. I sit on the EU board and the Scientific Advisory Committee to CPAP. More interestingly, perhaps, I'm not a Bayesian, but then I'm not a frequentist either. At least for today, what I want to do is to give a balanced introduction to the topic of Bayesian statistics in drug development, including its attractions, challenges, and opportunities. I have borrowed much of the material from lots of brilliant people and various resources, including Stephen Ruberg at Analytics Thinking. And for those who want to start a journey of reading, Stephen and a group of colleagues, including myself, recently published on Bayesian approaches in drug development in Nature Reviews Drug Discovery, and you can see the reference here on the slide. Here's a rough outline for the talk. I'll start with an overview of what we have been doing in terms of statistical methods in drug development and regulation, introduce Bayesian statistics and highlight some differences between the Bayesian approach and the frequentist approach, point towards various currently available regulatory guidance, as well as initiatives that are in the pipeline from the US FDA and from the European Medicines Agency, point towards some use cases of Bayesian statistics, and then give a summary. So let's start to think about what we have been doing in recent decades in terms of drug development, clinical trials, and medicines regulation. And this has been based on frequentist statistics. Everyone here knows, or at least we think we know, about p-values, about confidence intervals, etc. These are statistics that make statements about the data, the probability of observing the data, or something more extreme, given a prior belief. So we formulate a null hypothesis, perhaps that the drug doesn't work, and then we look to see whether the data that we generate in a clinical trial are sufficiently extreme to refute that null hypothesis. And the p-value makes a statement about the data in light of the null hypothesis. These statistics are based on what's called long-run properties. When we have a confidence interval, 95% confidence interval, let's say, we know that if we repeated that experiment multiple times, 95% of the confidence intervals that we generated would contain the true value for a treatment effect that we're trying to estimate. And again, the frequentist approach is to start with disbelief, a null hypothesis, perhaps that the drug doesn't work or that it's similar to a placebo. And we're able to change our belief and move to an alternative hypothesis that the drug does work or the drug is not a placebo if the data are sufficiently extreme. Probability of data conditional on a prior belief or given a prior belief. Now, what's this novel approach of Bayesian statistics? Perhaps it's not so novel. In fact, the Reverend Thomas Bayes wrote in response to an edition of The Doctrine of Chances. He replied or wrote an essay in 1763 stating, "The only thing I have is what I observe in natural phenomena. I must use the data I observe and infer what the state of nature is most likely to be true." This is reversing the formula or the metric I showed on the previous slide. Reverend Thomas Bayes started to talk about the probability of having a certain belief given the observation of data. And these are importantly different. The probability of data given a null hypothesis versus the probability of a certain belief, a treatment effect, or a null hypothesis given data. Remember, a p-value does not express the probability that the drug doesn't work. Having an extreme p-value 0.0001 doesn't mean there's 0.0001 probability the drug doesn't work. It's an observation, it's a measure of the strength and how extreme the data is, and how the probability that certain data or something more extreme would occur if the drug is not effective. There's other examples of this. The prosecutor's fallacy, for example, tells us that the probability of a person being innocent given the evidence is not the same as the probability of observing the evidence given a person's innocence. You can read about this online. To take a more medical example, think about diagnostics. A diagnostic test may have a false positive rate of just 1 in 1,000. So the probability of observing a positive test if the patient has no disease. This must not be understood as the probability of not having disease if you have a positive test. So these probability statements are importantly different. The way that frequentist and Bayesian statistics summarize information are importantly different. Bayesian statistics is founded on first deriving what's called prior information or prior distribution. This is an accumulation and an expression of current knowledge and beliefs about, say, a treatment effect. Having built and expressed this prior information, we then conduct an experiment, a clinical trial, and generate data. So results of a new experiment. We then integrate this prior information with the data and come up with what's called a posterior distribution, posterior information, the new best understanding of our treatment effect, updating our original knowledge and beliefs with the new data and expressing that probability of our new understanding in light of the new data. And what we'll do is take a look at 2 of the component parts here on the screen, the 2 new parts, if you like, the prior and the posterior. As I say, the prior distribution or prior information is an expression of what is known and what is not known, including an expression of uncertainties, based on any and all relevant and available data, and perhaps even based on opinions. We'll show some examples. This prior takes the form of a distribution. I'll show a number of plots like this, and you can think of the x-axis being a treatment effect, okay, and the y-axis essentially as a probability. So the plots are showing the current understanding of a treatment effect. And if we look at the top curve here, marked weakly informative prior, we see a very shallow sort of normal distribution. So this is saying our best understanding, which is where the probability is highest, is that the treatment has no effect. But I have a high degree of uncertainty about that information. I have a wide distribution of possible values going all the way past a negative treatment effect of -A and a positive treatment effect of +A. So I'm using a weakly informative prior centered on 0 to say, I really don't know anything about this treatment effect yet. It might be 0, that's my best guess, and I have a lot of uncertainty about where the truth lies. And if we move then to the bottom picture, strongly informative priors, we see 2 curves there. The curve on the left is showing a high belief in, let's say, a detrimental treatment effect. It's to the left of the point of no effect. Almost all of the distribution is to the left of the point of no effect. My prior belief tells me strongly that there's a detrimental effect of treatment that probably lies somewhere between -A and 0. Whereas on the right, the curve to the right-hand side is showing a strongly informative positive treatment effect. lying somewhere between 0 and B. So I'm using all of my available information and opinions to formulate what I currently know about treatment effect, including an expression of the uncertainty that exists in that knowledge and information. Here's some more examples, this time expressed on a risk ratio scale. So the point of 1 is where there's no difference between a treatment and a control, and you can see a blue curve expressing neutral information. So an a priori belief that the treatment effect between treatment and control might be similar. The green curve shifted to the right, this time for a detrimental effect, is saying, actually, I have information that says control is likely to be better than treatment. I might see a hazard ratio above 1 when I do my experiment. And then the orange curve, an enthusiastic or more positive prior, shifted to the left. A priori information and knowledge tells me my most likely estimate of the treatment effect is around a hazard ratio of 0.6, although you can see an expression of uncertainty because that orange distribution covers very extreme hazard ratios, both in a favorable sense and a detrimental effect going all the way out to hazard ratio 2. And this is the first key discussion point, perhaps, about use of Bayesian statistics. Who picks the prior and how? It must be a good thing in principle, right? Who can argue against transparently documenting current knowledge and uncertainties? Isn't it better to write that down before doing an experiment rather than being influenced and having sort of post hoc ideas around how plausible a certain treatment was in the first place? But we have to decide on the shape and the location of the prior. Do we start to assume a positive treatment effect, a neutral treatment effect, a negative treatment effect? How much uncertainty do we build in to the shape of the prior? So who should do this? Might the sponsor be too optimistic? Might the regulator be too cautious? Of course, there's other parties, but for the sake of simplicity, getting agreement between sponsor and regulators for every single time we want to do this in a clinical trial could be operationally challenging, just the sheer weight of difficult conversations that those two parties would need to have. And of course, within any party, any two individuals might have different perspectives on the strength of the available evidence. Now, we shouldn't criticize Bayesian statistics on this basis because it's still true after the fact. Okay, we're making decisions in a world where the sponsor and the regulator may have different views. Different individuals within a regulatory system or within a sponsor organization may have different views on the a priori strength of evidence. So these uncertainties already exist. Bayesian statistics forces us to write them down and have conversations about them in advance, which I think should be seen as a strength. Just a quick reference to certain methods that allow the data to pick the prior, so that more weight would be given to the prior information if the data eventually looks similar to the prior, and less weight would be given to the prior when data look dissimilar. So let's talk through a simplified example. Drug X has been developed in adults for a condition that also impacts children. Efficacy has been established in adults, perhaps with an estimate of X and a confidence interval of Y to Z. The disease in children is thought to be rather similar, though there are some factors that might impact Drug X's effect size in children. So we can go through a conversation about the biological, clinical, pharmacological understanding. We can look at the available evidence in adults. We can look at other drugs in that disease that have moved from adults to children and what relative effect sizes have been. And we can have a discussion as to what our best guess of estimate of efficacy in children would look like. Is X a good estimate? Is it too optimistic? Do we want to build in some uncertainty by choosing Y, the lower value of the confidence interval, rather than X, the estimate? How do we reflect other uncertainties about transitioning from adults to children, maturation, different concomitant medications, whatever it might be? by changing the shape and location of the prior distribution. We go through that systematic conversation and derive the prior distribution. And that might be expressed such as the blue curve on this slide. And now we move to the next part of the Bayesian approach. We conduct the experiment. In black, we see some observed data, and we put together the prior information and the observed data, and we derive the posterior distribution shown here in orange, now reflecting our best understanding of the treatment effect. And from this orange curve, we can make various statements. In black, we can express the probability that there's any treatment benefit, i.e. that the hazard ratio is on the right side of 1. In green, we can express the probability that the effect size is bigger than some clinically meaningful value, perhaps that's a hazard ratio of 0.85. Or in red, we can express the probability that the hazard ratio is more extreme than a competitor value, perhaps 0.8. And we can make a 95% credible interval for where the truth is likely to lie based on our posterior distribution. So whilst frequentist reporting is giving us estimates of effect size, confidence intervals, and p-values, Bayesian reporting is giving us an effect size, 95% credible intervals, and no statistical test. Instead, we get probability statements. And if we come back to our simplified example, we might take a strongly informative prior that there's highly likely to be some treatment effect in children. Available information might support that the probability of an ORR being greater than 20% is more than 80%, whereas the probability that that ORR rate is greater than 40% might be considered low, and similarly, the probability of the ORR being less than 10% might be considered low. And from that information, we can construct a prior distribution. We might observe 9 out of 30 objective responses in children, 30%, and we could calculate a frequentist confidence interval from that. But when we update our prior with the data, we can derive these posterior probability statements, the probability that the drug is effective in children given the data, the probability that the drug effect is greater than 20% in children, which might now move from 80% to 95%, for example, given the data we've observed. This leads us to the second key discussion point. And there's a distraction that we don't have time for. I asked the rhetorical question of how many points are in a game of tennis, because I don't know, and I don't think anybody knows. And similarly, how strong does evidence have to be to establish efficacy of a new drug? Yes, it's 2 adequate and well-controlled studies, but of course there's so many caveats to that in rare diseases, in oncology, hematology, when we're establishing treatment effects that can't ethically be replicated, etc., etc. And here comes the rub. If we incorporate exploratory data and make informative priors, so priors that predict there will be a positive treatment effect, it means we have a head start. Currently, we design phase 3 trials that start from 0, right? Treatment and control both start from 0, and we accumulate evidence that we eventually test. If we include an informative prior, the treatment group gets a head start. The treatment group is already ahead at the start of the trial. We would need to collect less information then in order to prove efficacy within the trial. Very simplistically illustrated by these 2 boxes. A regular phase 3 trial on the left, where all of our inference is based on the trial data. And on the right, a Bayesian approach with an informative prior, where some of our inference at the end of the trial, by design, is based on the knowledge we had at the start of the study. And we could choose then to generate less data during the trial. in my head, I conceptualize this using a sort of Roman facade. How many columns do you need to support the triangular top part of the facade? Is it called the pediment? How many columns do you need to support? And on the left, we see a depiction of a frequentist approach, where separate columns are built for the mechanism of action, the support we have from our understanding of the pharmacology, the support we have from our exploratory data, and then a big column support from our one or more phase 3 confirmatory studies. All of these providing independent pillars of support for a conclusion that the drug is effective. Whereas a Bayesian approach might work in a different way, starting off with the same ingredients, but gradually building evidence towards a conclusion of efficacy, integrating the information into one big column as the development program goes along. And with frequentist statistics, we all know where we are. One key strength as the frequentist framework, as it is currently applied, is to give clear standards and expectations. We run one or more confirmatory experiments that either succeed or fail with well understood consequences. But of course, this is also the weakness of the frequentist framework as currently applied, is that we dichotomize the results in this sense. If we have a borderline failure, p equals 0.055, I don't think anyone says that provides no information towards efficacy. But it is practice to dismiss that as a failed study. If we have 2 phase 3 studies, one reporting a significant p-value, one narrowly missing, is there sufficient evidence of efficacy? Or do we say one positive, one negative, you don't have 2 adequate and well-controlled positive studies? We might have 2 phase 3 programs where confirmatory trials are being run sequentially. The first trial starts with an unimpressive p-value, 0.4. In the other program, there's a borderline significant p-value, 0.06. Should we design the second studies in both programs the same way? The starting point, the information we have about efficacy from those first studies, even though they're both failed, is not the same. So there's an inherent weakness also in the way we apply the frequentist framework at the moment, which wouldn't be the same as we build this single column of evidence in a Bayesian framework, and we can take the strength of evidence even from failed experiments. But returning to this idea that an informative prior could allow us to collect less information, for some this is a disadvantage. And for other people, this is precisely the strength of a Bayesian approach. You might hear people talk about being able to derive the same strength of evidence from less information. And I think that's a good discussion point. Moving on to quickly scroll through some regulatory guidance documents. You can see reference to Bayesian statistics in E9, though not in glowing terms, perhaps. The document is open to the use of approaches other than frequentist statistics, though it does say when the reasons for their use are clear and when the resulting conclusions are sufficiently robust. It's a relatively old guidance document, of course, though I happen to think a very good one. There are newer guidance documents under ICH picking up on Bayesian statistics, pediatrics and extrapolation being a good example, and probably the adaptive designs ICH E20 that's currently under development. FDA are certainly active in this space. For a long time, for a decade or more, they've had guidance on the use of Bayesian statistics in medical device trials. And there is a plan by the end of year or financial year 2025, publishing draft guidance on the use of Bayesian methodology in trials for drugs and biologics. And it's not just writing guidance. The Centre for Clinical Trial innovation will partner with sponsors to integrate Bayesian analysis in parallel to frequentist analysis during the trial. So learning about the practical application of methods. And you can go and read a regulatory science impact story on how CDDF statisticians are using Bayesian hierarchical models to understand how drugs affect different groups of patients. And when you get the slides, these links should take you to the publications. In EMA, there's also activity. There's a document around complex clinical trials. This is more towards clinical trial authorizations, which touches on Bayesian methodology. And interestingly, the importance of adhering to or recognizing accepted regulatory approaches and clinical design techniques under the frequentist framework, also when you're designing a trial using Bayesian methodology. We'll touch on this slightly later. And the EMA methodology working party, their work plan includes a workshop on Bayesian statistics and the development of a reflection paper. There are multiple common use cases. So if you Google or if you read publications about where are people using Bayesian statistics in clinical trials and drug development, you might come up with a list like that on the slide here. Interim decision-making, dose response modeling, borrowing external information, of course, borrowing external information. Yeah, that's at the heart of the Bayesian thinking, the prior plus the trial data equals the posterior, yeah? And et cetera, et cetera. And of course, all of these things can be done with frequentist statistics too, and I think this is important to underline. For much of this conversation, in terms of the methods, we might have a choice as to whether we use Bayesian or frequentist approaches. And arguably, it's the mindset that's more importantly different than the methods. The explicit statement and integration of existing knowledge into statements about the probability of drug effects, a different mindset than the frequentist approach. And perhaps there are some areas where there are clear advantages to that mindset. Post-approval studies comes to mind. If we think about conditional marketing authorization in Europe, an MAH is required to complete a comprehensive data set post-approval and demonstrate that benefit risk remains positive. Some scenarios where a conditional MA is granted might be well suited to Bayesian thinking, updating our knowledge about drug effects, estimates, probability that the treatment effect is greater than 0 or greater than a clinically relevant value with those post-approval data, and hence evolving and updating the benefit risk assessment. Perhaps that approach, that mindset is even more suitable than judging whether a confirmatory trial fails or succeeds based on a p-value. Conceptually, perhaps it's more attractive to think about updating our knowledge in the post-approval space rather than managing or failing to prove to establish efficacy in a new study. And that might, I've put an example on the slide that you can read through, but the same thinking might be applicable to other post-approval work, variations or line extensions, for example. A couple of other topics before we conclude. I mentioned earlier that there's no testing. And of course, if there's no hypothesis testing, there's no hypothesis, not in the null and alternative sense, and there's no test, there's no statistical test, then in theory, there's no concept of a test result being an error. Type 1 error, type 2 errors, not applicable in Bayesian statistics, not relevant to the concepts. And in principle, no problem with multiple testing if we're not testing, right? But surely plays of chance in data can still mislead us away from the truth. Well, of course they can. Data can always mislead us from the truth just by chance. But data are the Bayesian's truth. There's some interesting emerging sort of hybrid approaches, if you like, where Bayesian statistics incorporating some frequentist principles and standards are being used. So whilst we might not adjusts for type 1 error control and multiple testing in a trial with interim analysis under a Bayesian framework, we might require a higher posterior probability of a treatment effect in order to stop a trial at an interim analysis. Another issue to point to is the investigation of subgroups. It's a notoriously difficult problem, of course. Does a subgroup result reflect a true effect modification or a play of chance? Well, there's an EMA guidance document. I'm biased, but I happen to think a very good one, that tells us to look at all available evidence to help determine credibility. Think about the biological plausibility for the effect modification that's been observed. Look for evidence of replication that effect modification has or has not been observed in other data sources. Well, if we do that in advance, we're essentially being Bayesian. If we're building that information into our prior, we're essentially being Bayesian. And there's not time to go into this today, but look into the use of Bayesian hierarchical models, levels of trial, subgroup, patient level data, etc., where we can build in separate priors and understandings, expressing our current knowledge about a range of treatment effects, not just an overall treatment effect, which might work well in the investigation of subgroups. Maybe we'll come back to this a little bit in the Q&A session. You can also read about the BATTLE study, which used this Bayesian hierarchical modeling, essentially along with adaptive randomization, to push patients towards treatments that were more likely to be effective for their particular type of lung cancer. To sum up, Bayesian statistics are sort of intuitively attractive because they make direct statements and inferences about drug effects, not about data. So we use our prior, we use our data to develop this posterior probability statement around the likelihood of there being a drug effect in light of everything we know, rather than p-values making statements about data. Specification of a prior is a strength, writing down, stating explicitly what it is we think we already know. But it is perhaps the big operational challenge with Bayesian statistics. Using informative priors gives us the possibility to generate less information. And in some settings, it will make very good sense. But of course, when it does make sense to do so, is a policy decision. And in my opinion, at least, requires policy level discussions about when we want to generate less information to support regulatory conclusions. It shouldn't just evolve because we choose a particular statistical approach. Arguably, Bayesian statistics is giving us a better use of data and patients in some circumstances, at least compared to the way that frequentist statistics is currently applied. We can use information from failed studies explicitly. It's an intuitive way to accumulate evidence and to bridge between populations and experimental settings where they're related. I want to come back to this idea of Bayesian methods and Bayesian mindset. We can use Bayesian methods just as well as frequentist methods to perform some of our clinical trial work. And you can turn that around. We can use frequentist methods just as well as Bayesian methods to perform some of our clinical trial work. The big change here is in the mindset, the Bayesian mindset of integrating our prior information in order to draw conclusions. And there is this renewed momentum at regulatory agencies, both sides of the Atlantic, ICH, so that guidance can be developed on how Bayesian and frequentist approaches can exist in harmony. I hope it was interesting. I'm looking forward to the discussion section.
Thanks a lot, Rob. This was excellent. It was a very interesting and, well, thought-provoking presentation. At least from my side, being a CHMP member, I sort of foresee that we gradually we'll see some more use of Bayesian methods, and that might become challenging. We have a few questions in the Q&A, and I have a couple of questions myself, of course. But let's start at the top with a question, a more technical question. Why did you represent the prior as a Gaussian distribution? Is the first question I see here.
Yeah, thanks, Even, and thanks for the question. So, yes, on a number of the slides representing the prior distributions or other distributions during the talk, we saw this normal type distribution, this Gaussian type distribution. And when that's being used to represent a prior, what we're saying is our best understanding of a treatment effect based on the existing information is at the center of that distribution. And we're sort of equally uncertain about the treatment effect as it moves away from that best estimate towards a more positive effect and towards a more negative effect, with a sort of gradually reducing probability that these extreme values are the truth. So we end up with a distribution that's centered around our best understanding, with gradually reducing probability for stronger or weaker effects, and hence a normal or Gaussian distribution.
Thank you. I guess that was helpful. Let's pick another one. Based on your presentation, Bayesian statistics in its utilization of prior knowledge makes total sense. So how did frequentist statistics dominate over the past 2 centuries or so, and why? Is it because it's simpler to conduct?
I don't have a good answer, really, for this, to be honest, because, as you say, this sort of the establishment of frequentist statistics sort of predates my time being educated at university in statistics, where I learned more about p-values and confidence intervals than I did about priors and posteriors and predictive probabilities, etc. And so it predates my education, and my experience in the industry has been predominantly with frequentist statistics. It is true that the integration of a prior distribution along with the data that you observe, or the likelihood, can be mathematically more complicated, and that has limited the use of Bayesian statistics when computational power was less. I understand from those practicing in the field that this is almost a negligible problem most of the time in today's world. But this is, I don't know which country you're asking the question from, but it brings to mind in the UK the competition we had between VHS videotapes and Betamax videotapes. And it wasn't that one was better than the other. One was just more successful in its marketing and became the norm and killed off the other.
I think you're correct in assuming it has to do with computational power. I guess it would definitely be easier to be a frequentist 50 years ago, and it has become easier to be a Bayesian statistician nowadays with computer power. That's my guess, at least. And then another one, what if there is no prior at all, if there is no prior information?
So that's good. And sorry if that wasn't clear from the talk, because I guess one answer to this is there's never no prior. We always have some information by the time we get to the stage of doing clinical trials. Okay, there's always, even if it's only a scientific rationale that's well founded by good understanding of biology and chemistry, etc. But there are times, perhaps many times, when we want to use weakly informative, or sometimes they're referred to as non-informative priors. And so this distribution, let's talk about the Gaussian normal distribution again, would be centered around 0 and would be extremely wide, reflecting a high degree of uncertainty. And essentially, that's how a prior reflects absence of information. Centered around 0, extremely wide in terms of the possible treatment effects.
Thanks. I guess that answers that question. Do you want to pose a question, maybe, Axel? Ah, you're muted.
Sorry, I had some difficulty in unmuting. Let's take the next one from the list here. Is it fair to say, this attendee asked, that Bayesian statistics is intrinsically more complicated than frequency stats? So you talked about the computational part. For me, as a non-statistician, I felt that, but that may also be the way I've been brought up, that frequentist statistic is just easier. It's, you know, there is a fallacy of considering it delivering binary results. And maybe one reason that the non-statistical community prefers it at the time being is that it serves this need of answering whether it's true or false. And the Bayesian way of thinking about probabilities may be closer to reality, but it may be more complicated or more or less intuitive. What do you think, Rob?
I think there's a couple of points to make. And one I tried to make in the talk, which is part of the complexity that we're pointing to here, is the need to specify the prior in quantitative terms. So Eva sits at CHMP, I used to sit at CHMP, and eventually you have to make a decision on whether a product should be approved or not. Do that by looking at phase 2 data, phase 3 data. You have in mind what the mechanism of action is, etc., etc. And your mind puts all of these pieces of information together and you decide if you're convinced or not. We don't write down all of that information before the phase 3 is into a prior, into a quantitative form. Okay, people make their decisions. Bayesian statistics is forcing us to do that and to specify our prior, and that is difficult. So that is a source of complexity. It's not necessarily a bad thing just because it's complicated, right? So that's one point. And then there is this computational point that we referred to earlier. So the need to integrate the prior, which may have one form, with the data, which may have another shape and form in terms of its distribution. And that can be complicated. Again, it's not the wrong thing to do, or it's not a bad thing just because it's complicated. But yes, there's those elements that are more complicated than implementing a frequentist approach, where we're looking at the data from each experiment sort of in isolation before doing that integration part when it comes to decision-making time.
Thank you.
Yeah, can I maybe follow a little bit up on that? I think from my perspective, my biggest worry is that we tend to want to use Bayesian methods for, well, small trials to have less data, but that will mean a lot of weight might be put on the prior. And if you put a lot of weight on the prior and you actually make that prior yourself, it will affect the result. And I think from my perspective as a regulator, I would be very worried that the prior would sort of drive the results too much. Do you have any comment on that?
And of course, the Bayesian would say, if we're certain about the prior and we've got good justification, it should drive the results, because it is knowledge that's already established that I've just written down in quantitative form. Of course, a lot of the times when we're doing clinical trials, we're doing them because there is uncertainty. So I briefly described in the talk a sort of a case study of an extrapolation exercise where a drug has already been shown to be effective in adults, and we want to go and work out, we want to go and investigate whether the drug will also work in children. We may not be able to do a large RCT in children. And if we have good information in adults and good information that disease is similar between adults and children and that the drug will work the same way, we may not need to do a large study, comparative RCT type study in children and start from zero. You can even argue that that's unethical, not to leverage the information we've already got in adults. But now we get to Eva's good question and comment. How strong, how much weight should we put on that adult data? And when we were writing the extrapolation guideline when I was at EMA, we included some text in this idea that the data generated in the target population, so children in this case, should be the data that dominate. So the implication being that the information you borrow from adults, the weight of that would be less than 50% of your final answer, if that makes sense. But of course, this should be case-by-case discussion, and sometimes there's more uncertainty where you would want to give the adult data in that scenario less weight by having a less certainty in your prior distribution, wider tails rather than a sharper, more tightly contained narrow distribution. And there'll be other times where you want to put more weight on the data from the source population, the adults in that case. And this is to exemplify, if you like, how detailed each and every conversation would need to be. Again, that doesn't make it wrong to do so. It just, but it does put a lot of burden on because those conversations are important.
Yeah, maybe as a follow-up on that, there is a question in the Q&A. If we start to use Bayesian designed phase 3 trials, will this cause some kind of challenge for cross-trial comparison?
Okay. So perhaps the question comes from the idea that if a historical phase 3 study is summarized by a estimate p-value and confidence interval, and a trial with a Bayesian design is summarized with a posterior probability statement and a credible interval, I might not be able to make those comparisons, or I might not be able to compare A with B. And that's a good thought, because again, we've got one statement about the probability of the data under a null hypothesis versus the other, the probability of a treatment effect given the data, and we can't compare those two probability statements in a naive way. However, we've still got the data that have been generated in those two experiments. And so whether using a frequentist approach or a Bayesian approach to make a proper between study comparison, we would still be able to do that. Just making sure we use the same metrics and the same methods to make that between study comparison. So not a naive comparison of the outcomes, but absolutely we can still compare the data from the two studies using an appropriate method.
Good. The next question is a bit more technical. It says the modified toxicity probability interval design seems to be a default method for phase 1 dose finding studies. Could you elaborate a bit on that? And with an eye to the clock, I would say let's try to keep the answers brief. We have a lot of open questions and the time is advancing.
So I could be fairly short with this one. There's been a number of model-based designs, model-based approaches to phase 1 dose response type trials, moving away from the old 3+3 designs. A number of those novel ideas have used a Bayesian framework for their implementation, and this tends to work very well and be very sound methodologically.
Okay, thanks. The next question is, have you seen some change in the mindset, in quotation mark, of clinicians and regulatory agencies to accept Bayesian methods? I think you have partly answered that already, but maybe you can round it up.
And maybe I should have made this clearer in the talk as well. Bayesian methods are in use. Companies are using them for their internal decision making. Regulators are approving clinical trials that use Bayesian methods for interim decision making. For example, a company may use a Bayesian approach as part of their investigation of subgroup data once the trial results are in hand. There's lots of different ideas behind Bayesian, or lots of different uses for Bayesian methods, and they are being used, they are being seen by regulators. The gold medal, the big ticket item here would be a whole development program where you sequentially generate and then incorporate data into a prior that leads you to a strongly informative prior for a phase 3 trial, which is then smaller, or potentially smaller in size, because you've already got all this prior information. And that scenario is, I think, one that's not yet tried and tested or practiced, and there's been a lot of exposure to, and the regulators would quite rightly have a lot of questions and considerations about. And I'm sure this is sort of actively under consideration as regulators develop these guidance documents that might appear over the coming years.
Thanks. The next question here is, there may not be hypothesis testing in Bayesian design and analysis, but multiple claims can be made on one Bayesian trial. How to deal with a false discovery problem if it is not classical FWER control in frequentist framework? Is there regulatory consideration?
So I don't know if now is not the time to get into the philosophical part of this, I suppose. The practical answer, as I said in the talk, is yes. That must be a consideration for a regulator. You know, if I put myself back in that seat, I think about multiple potential claims being generated from one data set. I can't help but be concerned because, as I say, data can mislead a Bayesian from the truth just as much as it can a frequentist from the truth. The Bayesian paradigm doesn't have this concern with multiple testing in the absence of tests, and the data are the best available information for any given claim, right? But Eva, you might want to comment yourself, but I would think the regulators will still be concerned with the possibility of being misled by testing multiple claims within one data set. And as I say, it's led to some of these hybrid approaches with Bayesian summary statistics, probability statements about drug effects, but where the required strength of evidence is adjusted for repeated generation of probability statements. So instead of wanting a posterior probability greater than 97.5%, let's say, to be convinced of a drug effect, we want to see probability of drug effect greater than 99% because I'm testing it 2 or 3 times. Now, that doesn't really fit with the Bayesian mindset and paradigm, to be honest, but it's a sort of practical trade-off to be using the method whilst addressing some concerns. And I think this is a sort of temporary compromise, yeah, as the two worlds collide, which is another thing I guess in the end regulators would have to take a position in guidance documents.
Yeah, I don't think we have taken a firm position yet, but of course we would be worried about multiple claims from one single data set. So I would tend to agree with you on that. The next question on the list here is Bayesian methodologies have been recommended in first-in-human phase 1 trials. And based on simulations, the claim has been made that the RP2D is better defined compared to rule-based models. Would you agree with this, that this claim should be substantiated with a randomized phase 1 design? Bayesian versus rule-based setting?
Should we understand rule-based to be similar to the 3+3 notion of escalate if I see this and don't escalate if I see that? Yeah. No, absolutely. Yeah. This has been known for 20 years or more, the deficiency with the 3+3 design. It's gobsmacking how it still survives to a methodologist, yeah, to someone on the outside. Maybe there's very good clinical or operational reasons in some cases, but no, absolutely. The model-based approaches, which can be frequentist, are often Bayesian, will outperform that type of fixed 3+3 design and its decision rules.
And the next one is, are there differences in how a Bayesian approach would work for assessment of efficacy versus assessment of safety?
Well, that's a good one. That's a good one, partly because it really calls into question what we do with safety data. Anyway, you know, we don't tend to be frequentist or Bayesian with our safety data as a rule. We just tend to be descriptive and to try and understand. There are exceptions, of course, but as a rule, we just tend to describe safety data. And when we do make comparisons between groups, honestly, those comparisons are often flawed. Longer exposure time on treatment group than on control group, for example. So where we are needing to make formal comparisons, the merits, the pros, the cons, the challenges, the opportunities of the Bayesian, I would say, are similar between efficacy variables and safety variables. There's nothing that makes it less relevant to consider a Bayesian approach when you're talking about a safety variable than an efficacy variable, if it's one where you want to make a formal comparison. At least nothing that comes to my mind.
Thank you. Do we need to assign a weighting, here is the next question, to the a priori knowledge as we included in the statistical analysis?
Yes, and that's what we're doing as we develop our prior, just to try and make it simple. We might have two prior distributions which are located at plus X as our best guess for the treatment effect. Okay. If we have a narrow distribution, which goes from X over 2 to 2X, so it's not Gaussian, but okay, narrow distribution, that's saying we've got quite a lot of certainty in our understanding. Whereas if we have a broad distribution that goes all the way into negative values and all the way up to 10x or 20x, says we have a lot of uncertainty. And we can and should weight the prior, and this is back to Eva's question earlier, as to how much we want the prior to contribute and how much we want the data that we're going to generate in our next experiment to contribute. The more we want to rely on the data rather than the prior, perhaps because of uncertainty, the more uncertainty will reflect in the prior distribution.
And the data doesn't have to be clinical all the time. It can be modeling and simulation. That's another question asked here.
And that's good in terms of the contribution to the prior. There's all sorts of inputs that could go in, including surveying expert opinion as to what a likely treatment effect or potential range of treatment effects might be. Now, you might say that, okay, if that's the way you need to form your prior, perhaps you should be thinking about a non-informative or weakly informative prior instead, or at least you should reflect that opinion with a great deal of uncertainty and allow the data that you're going to generate to dominate. But yes, Axel, lots of different sources of information can go into constructing a prior.
A follow-up on that is perhaps not the next one, but the one after that. It would seem we are generally quite uncertain about the prior, or why are we doing the study?
So say that again.
It would seem we are generally quite uncertain about the prior, or why are we doing the study? I mean, I think the one questioning this is saying, if we know the prior, why on earth do we want to do the study at all?
I mean, that's a fair question, isn't it? That if you have great deal of certainty about the prior, maybe you wouldn't do the study. So let's think about a study in a common disease, and this still happens, where we might have 3 pivotal studies all positive. You could go and construct a prior for your 4th pivotal study, and that prior would tell you don't bother doing another study. You've already got such certainty about your results. Yes, it's true. Most of the time, we're still doing research and we're still sort of in equipoise. We all know the problems with that conversation, but we're still sort of in equipoise, despite the fact that we've got existing information and we're still able to randomize to placebo under certain criteria or whatsoever else, or randomize against standard of care in oncology, hematology settings, despite having some promising data already.
Thanks for mentioning the equipoise. That was on my mind too. And I think the clear process of defining priors helps to see whether equipoise is still available. Yeah, the next question is an operational one. Would you recommend to get consultation advice from the EMA to define the prior?
Yeah, of course. And I'm not just saying that because Eva's here or because of my history, but there's a line of argument that says, go and generate the data, go and generate lots of it, and then everyone can apply their own prior. And if a company wants to be more optimistic and a regulator wants to be more pessimistic, they can apply their own prior to the data. Okay? But of course, then the company is left thinking, well, how much data will I have to generate? How skeptical will the regulators prior be that I have to overcome with the data I'm going to generate? So you still need to know what are the success criteria going to be, and therefore how much data am I going to have to generate to meet those success criteria? And until there are sort of established practices in this space, then yes, we'll have to have those conversations one by one with regulatory agencies all around the world to get their view.
The next question is actually a nice follow-on on that. Do you foresee regulatory approvals based on pivotal studies using Bayesian probabilities as evidence rather than frequentist p-values? And if so, how far may that be in the future?
Look, I don't know that it needs to be very far at all because, and again, I sort of speak under the control of Eva with being the CHMP member, but the regulator approves the drug, not the trial or the method. Yeah. And we've all seen trials and development programs where it is blindingly obvious that the drug works. Yeah. And if that was established through frequentist statistics or Bayesian statistics, the evidence would still be overwhelmingly strong, such that it would be very hard for a regulator to write a major objection or a ground for refusal saying, well, you've used Bayesian rather than frequentist methods. Yeah, that doesn't work. As ICHE9 said, you still have to prove the evidence is sufficiently strong. But in practical terms, and to make this more day to day and sort of standard practice, I think we're looking forward to receiving whatever regulatory guidance and testing methods through interactions with FDA, through interactions with EMA, and I imagine it's still a number of years before we get established guidance documents and get an idea for what this might look like, and then it would have to be implemented in trials. So in practical terms, it's not going to be in the next year or two that this becomes the norm, but there is a keen interest from industry, I would say, and there is an openness from regulators to start having these conversations and doing this hard work to think about what the application of the framework or the paradigm would look like.
Thank you very much. Eva, would you like to take the last question?
Yeah, I can quickly also comment. Maybe I wouldn't mind seeing a Bayesian designed trial if I wasn't worried about the prior. If I was worried that the prior would be too optimistic and too much weight is put on it, I might be a bit skeptical. But if the prior wasn't too informative, I'd be happy, even if I was brought up as a frequentist originally.
And Eva, sorry, just before we go to the last one, just 15 seconds, because I think the other challenge for the regulator, as I see it, is that the way we set success criteria for pivotal studies at the moment, whether it's one or multiple pivotal studies, is to say, we already know, and this was the idea of the Roman pillars, we already have an understanding of the mechanism. We can already look at your exploratory data. So the amount of information you now need to provide to convince us is represented by statistically significant tests at the 5% level. If we've taken our mechanism of action and built a prior for phase 2, and then taken our phase 2 results and built an informative prior for phase 3, it's not necessarily the same to say that your prior plus data from your smaller phase 3 study should meet a posterior probability that's equivalent to a p-value of 5%. The regulator now has less information than they did before because we don't have that summation across the component parts. They've all been integrated. So it's the development of the prior and it's the discussion of the success criteria that at least to me, it makes sense still to have on the table. And we don't have to live in the past when it comes to defining what those success criteria are, because the world looks very different now with rare diseases, genetically defined diseases, different types of products, stronger mechanisms of action, etc. But we do have to have that conversation.
Yeah, and this points to one of your slides with the pillars, with the sort of the house and the arrow, which I really liked a lot. I think that was illustrative. Yeah, the last question is From one saying, I'm a bit concerned that the assumption is made that without Bayes, not all information would be collected and considered and modeled into decision or molded into decision making in clinical trials. The consideration of the totality of data is just professional work done by every PI or research group. Do you have any comment to that?
Yeah, I completely agree. The idea isn't that the Frequentis world dismisses all of that information. That's not the point at all. Absolutely not. And again, that's how regulatory systems have been working now. Yeah, and researchers, academic institutions, etc., etc. The point is only to illustrate the difference in the Bayesian paradigm, where we take all of our existing knowledge and explicitly quantify that as our prior distribution. So it's a different way of working with all of these multiple pieces of information. And of course, even in the Bayesian world, we have our prior, we have our experiments, so we can integrate those and generate our posterior understanding. But at the same time as doing all that, there can be other pieces of information, other sources, competitor development programs, whatever it might be. The science moves on. And so there's always more information to integrate. We can't turn our brains off at any point. So that really wasn't the point that the current world ignores information. It just doesn't write it down in this explicit, transparent way that is intended by the Bayesian prior.
Thanks a lot. Eva, any final comments from your side?
No, just a big thanks to Rob for doing this. And thanks to everyone posing questions. I think we had a good discussion. Hopefully people had answers they could actually understand, I guess. Thanks anyway.
Everything will be on our website in a couple of days, so if you want to repeat it, you'll find it there. Many thanks also from me to Rob for a complex topic, very well explained, and many thanks to you, Eva, for joining me in this. So I have a few final slides, and also I want to tell you about how to get a certificate of attendance. So first, upcoming events, we have a session at ESMO for the first time, a joint session of ESMO and CDDF about regulatory challenges in clinical cancer drug development at the ESMO meeting in September. And then we have a joint workshop with the ERTC. It's a multi-stakeholder workshop held in Amsterdam on the 23rd and 24th of September. You can join in person or online. Next slide, please. 2 more webinars, one on advances in artificial intelligence, generative AI to support regulatory submission. We will hear from Katrin Rupala, head of regulatory at J&J. And then we have a very exciting talk as well about a hot topic, EU clinical trial regulations on the 12th of November in another webinar, and you know already how to sign up for these. So thank you very much. The last slide shows you how to get a certificate of participation, and very important for us, please fill out the survey form to give us feedback. I would like to thank everyone again and wish you a very nice evening. Goodbye.